What are anchoring junctions and how do they work? Now, the specific name for anchoring junctions are going to be either desmosome or hemidesmosomes. And so desmosomes are going to be located between two adjacent cells, and we can see kind of a complex formed by the proteins here that are going to be anchored to the cytoplasm. So kind of coupling the cell together kind of mechanically and structurally. So we're going to hold the cell together, these two cells together. And so we've got some extracellular components here that again are going to bind these desmosomes together. And then we are going to anchor that next cell through these protein components into its cytoplasm as well. And so this desmosome then is basically going to form this structure that is going to tightly adhere one cell to the neighboring cell. In contrast, a hemidesmosome, hemi for half, basically means we've got half of that desmosome structure. So we've got the desmosome structure within one cell, the proteins here, the protein kind of fibers kind of extending out into the cytoskeleton, anchoring the entire cell and cell cytoskeletal elements, the entire cell. In this case, the hemidesmosome is not going to go to another cell. It's going to go to the connective tissue. And so it's going to then kind of penetrate through the basement membrane and connect with fibers within the connective tissue. If we look at this in a little bit more detail, what we're going to see is that these desmosomes right here, kind of magnifying it up a little bit, they're described as having this kind of dense appearance. They're going to have a plaque formation. We've got intermediate filaments that are going to extend from the desmosome into the rest of the cytoskeleton. And so we're basically holding the entire cell together with this desmosome boundary. So we're linking the cells together. We're linking the cytoskeleton of these cells together. And this is going to result in these cells being held together in a coherent sheet. In addition to that, we've got some extracellular proteins here. They're illustrated by these linker proteins, an example of which is a cadherin. This is a cell adhesion molecule that's involved with, again, helping one cell kind of connect up with the neighboring cell. We take a look at the hemidesmosome down here at the bottom. You see the same basic structure. We're going to use a different linker protein. In this case, we're going to use an integrin, and this integrin then is going to be able to bind to some of the glycoproteins that are located within the extracellular matrix of the connective tissue. In both of these cases, we've got these spot-like connections that are going to be coupling these cells together, or in the case of a hemidesmosome, coupling the cell to its surrounding connective tissue. And so we're basically going to be holding these cells in place structurally.